So this is the molecular genetics unit, and basically we're talking about DNA, how DNA codes for proteins, and some of the technologies that we use now regarding DNA. So in the molecular genetics unit, we are studying DNA, and we're studying how DNA codes for proteins. And we're also going to talk about different technologies related to DNA. But first, we're going to talk about how DNA relates to evolution. And we've already indicated that commonalities between two different species indicates a common ancestor. So let's take a look at what DNA and the proteins that it codes for indicates about evolution. We've already talked about DNA, and we've said that DNA is made of 4 different bases, and the fact that all organisms utilize the same 4 bases in their genetic makeup indicates that there is a common ancestor between all organisms on Earth. Now, we can use the similarity in base sequences to analyze just how related 2 different organisms are. So, for example, we're finding new species all the time. Now we can sequence the DNA of that new species and see what other species here on Earth it is most related to. Now let's talk about proteins and the information that they offer about evolution. Now, if you remember, DNA codes for mRNA, and mRNA codes for the amino acids that make up proteins. If you remember, we divide the mRNA into codons, and there's 3 bases to 1 codon. Now, this is what I mean by the triplet code. So a triplet code would be like AUG. That would be a codon found on mRNA. And if you remember, AUG was the start codon, and it always coded for the same amino acid, MET. So MET is the amino acid. Now, the fact that all organisms follow the same triplet code, so a AUG in a human cell is going to code for methionine, just like AUG in a yeast cell is going to code for methionine. So that codon chart that I'm going to show you here in a little bit, it is the same for all organisms on Earth, which indicates there is a common ancestor. Now, when we build our proteins out of the exact same 20 amino acids. Again, evidence of a common ancestor. Now, I just described to you a little bit about protein synthesis and the fact that DNA gets converted into mRNA, and the mRNA gets translated into an amino acid sequence. And you probably remember that from the biochemistry unit or from pre-AP when we talked about protein synthesis. This flow of information from DNA to RNA to a protein has a term. It's called the central dogma. And if you think that's a weird term, I agree with you. But anytime someone is talking about, or you're reading about something that says the central dogma, then they're basically talking about the steps of protein synthesis, transcription and translation. And the fact that all organisms have the same flow of information from DNA to the mRNA to the amino acid sequence in a protein indicates a common ancestor. So all organisms go through transcription and translation in the making of a protein. Now, just like we can sequence DNA and figure out the order of bases and use that to determine genetic relatedness, we also can sequence the amino acids in a protein, and that indicates how related 2 different organisms are. Now, there is a protein that we can find in all organisms. As a matter of fact, we just talked about it, even though we didn't name it, because we were talking about proteins and the electron transport chains. Well, one of those proteins is called cytochrome C. So let's go to that mitochondria, and let's draw on the inner membrane, and let's put some of those electron transport chain proteins in there. And one of them is called cytochrome C. And we find cytochrome C in all organisms, even bacteria. Because remember, bacteria, they also have an electron transport chain. We find that they'll have that cell membrane and that cell wall, and that makes a space where they can concentrate protons. So they also have cytochrome C. It's embedded in their plasma membrane. Now, we can sequence the cytochrome C, determine the order of the amino acids, and that gives us an idea of how related 2 different species are. So I put this chart here because I think it's pretty fascinating that when we sequence our amino acids in cytochrome C, and then we sequence other organisms, here's some of the commonalities that we find. That us and chimpanzees have the exact same amino acid sequence in our cytochrome C. If you compare us to a rhesus monkey, all of our amino acids are in the exact same order, except there's one difference. If you compare us to a rabbit, there's 9 differences in the amino acids of cytochrome C. And it tells us just which species we're most related to. What we're going to focus on next is a little bit of history about DNA and how we came to know about DNA, and that it was the genetic information and not proteins or carbs or lipids that were getting passed down from generation to generation. And we're also going to learn a bit about the history of the discovery of the structure of DNA. And how did we know that it has 4 bases, A's and T's and G's and C's? And how did we know that it was double-stranded with a phosphate sugar backbone? So you're going to see that as we talk about the different findings of these scientists in relation to DNA, you're going to see that the research of one contributed to the research of others. And that's an important idea about science. And it's important that we publish our findings and that they're good, accurate findings, because other people are going to analyze our findings, and they're also going to use that information to further their own research. Now, in this big, thick curriculum document that I get from College Board, it tells me that you need to know these scientists and how they contributed to our ideas of DNA. So I'm just going to tell you, you need to memorize the names, you need to memorize what they did. So we're going to go through these different scientists, and we're going to take a look at these experiments they did and the data that they found to see if we can draw conclusions similar to what they did. So first we're going to look at the research of Frederick Griffith. He was set out to prove that there was a molecule that was actually coding for traits in organisms. And then later on some other scientists figured out exactly which molecule it was, whether it was the protein, carbs, lipids, or nucleic acids. So here's what Frederick Griffith did. He was working with strains of bacteria. And there is the R strain, and then there's the S strain of bacteria. Well, the R strain is non-lethal. You can take this bacteria and you can inject it into the mouse, and the mouse is going to be just fine. But if you take this smooth strain and you inject it into the mouse, then the mouse dies. So he decided he was going to denature or kill this lethal smooth strain and inject it into the mouse. So he heat killed those lethal bacteria cells, injected them into the mouse, and as you can probably imagine, the mouse lived because he killed those bacteria. Well, then what he did is he killed the deadly bacteria, but then he mixed them with some living R strains. So that's why we see both of them here in this diagram. And what he found is whenever he injected this combination of alive, non-lethal bacteria and dead lethal bacteria, for some reason the mouse dies. So here's what his findings suggest, is that there was a molecule that was being transferred from these lethal S strain bacteria to these non-lethal rough strain bacteria, giving them a trait. So they were now deadly to this mouse. So there was a molecule, so let's indicate that. There was a molecule that was being transferred from the smooth strain to the rough strain, making those rough strain now lethal. So there is a molecule that is giving organisms and cells their traits. Now the next group of scientists, Avery, McLean, and McCarty, they set out to figure out what molecule is it. Is it the proteins, carbs, lipids, and nucleic acids that are being transferred from those deadly bacteria cells to the non-lethal, giving them that new trait? So when they were done with their experiment, they discovered that it's DNA that is the molecule that is the genetic information of organisms. And they had pretty much figured out it wasn't going to be the lipids or the carbohydrates that were coding for traits, but they weren't sure if it was the DNA or if it was proteins or RNA. So they set out to figure out which one is the genetic information of organisms. So here's what they did, is they heat killed these S cells. And remember, these S cells are deadly, or they're virulent, means deadly. And they took out the DNA, RNA and proteins from these deadly cells. And then they mixed them with these non-lethal R bacteria cells to see which of these molecules would cause these R cells to become lethal. So in this mixture, they had the DNA, they had RNA, and they had proteins. And what they did is they had 3 different samples of this mixture of molecules. And if they add proteinases, hopefully you understand those are going to be enzymes that are going to chop up proteins, destroy proteins. So basically this sample did not have any protein in it. And whenever they added these R cells, then they ended up with these deadly R cells again. So something was in that mixture that was being taken up by these non-lethal cells, making them now deadly. So since there was no protein in there, it could not be the protein. So protein is not the genetic information, because it was gone, yet these cells still were able to become lethal. So in this other sample, they put an enzyme in there, and that enzyme basically chopped up the RNA. So the sample does not have any RNA. So whenever they put these R cells, again, these R cells were non-lethal, into this mixture, they became deadly. So since this sample did not contain RNA, then it could not be the RNA that was causing them to become deadly. So again, RNA is not the genetic information. So in this third sample, they put enzymes that are going to break down the DNA. So the sample does not have DNA. When they added these R cells, then these R cells did not become deadly. So this proves that DNA is the molecule that must be in the sample and is giving these bacteria cells their traits. So that's the big idea that was gained from the research by Avery, McLean, and McCarty. Now, around that same time, there was 2 scientists named Hershey and Chase, and they were also trying to prove which molecule was the genetic material of organisms. Now, in their research, they were using viruses, and these viruses target bacteria cells, which I know sounds strange, but even bacteria cells can be infected by viruses. And viruses that can infect bacteria cells are called bacteriophages. And we're going to be talking more about these guys later on in the unit. Now, bacteriophages are very simple structures. They're basically made of 2 different molecules. They're made of proteins and DNA. So Hershey and Chase set up 2 different treatments. So here's treatment 1. Here's treatment 2. So to summarize what they were trying to do is they wanted to tag the proteins and watch where the proteins go whenever these viruses are mixed with these bacteria cells. And then in another treatment, they wanted to tag the DNA and watch where the DNA goes whenever these bacteriophages are mixed with the bacteria cell. So they wanted to basically see what it was, what molecule the viruses were using to infect these bacteria cells. So in the first treatment, they tagged the DNA, and they used a radioactive isotope. They used these phosphorus atoms that were radioactive. Now, the reason why they use phosphorus atoms is because of something I've had you memorize in the biochemistry unit. Nucleic acids contain carbon, hydrogen, oxygen, phosphorus, and nitrogen. So there's these phosphorus atoms that we can tag. And the important thing to note is that proteins do not contain phosphorus atoms. They contain carbon, hydrogen, oxygen, and nitrogen. So we will not accidentally also label proteins when we're trying to label these nucleic acids, because they do not have phosphorus atoms in them to tag. So here's what we saw in treatment 1. We tagged the DNA that's inside this part of the virus. We infected these bacteria cells, and then we basically separated the viruses from the bacteria cells. And this is where we found the tags. We found these 32P phosphorus tags inside the bacteria cells, indicating that the DNA is what is being injected into these bacteria cells, and the DNA is the genetic material of these viruses. So let's take a look at treatment 2. In treatment 2, they use these radioactive sulfur atoms to tag proteins. Now, if you look up here, you can see that there's no sulfur in DNA, but there is some sulfur atoms in amino acids that make up proteins, specifically cysteine. So I'm going to indicate cysteine is the amino acids that contain sulfur. So we were labeling these proteins so that we can track where they're going to go. So again, we infected the bacteria with these bacteriophages, and we separated them, and this is where we found the tags. We found these 35 sulfur tags outside of these bacteria cells. So it indicates that protein is not the genetic information that viruses are using to infect these bacteria cells. So next we're going to look at the findings of Watson and Crick and Wilkins and Franklin. And the reason why they're grouped like that is because Watson and Crick were working together in one lab, and Wilkins and Franklin were together in a different lab. And Rosalind Franklin was using X-rays to produce pictures of DNA. And she took this famous photo right here. This is called photo 51. And though it's kind of hard to tell, basically what this photo shows us is that there's 2 strands to DNA. DNA is double-stranded. And it is in a double helix. So we see one strand of nucleotides coming down this way. And then we see the other strand kind of coming this way. And if you look at a picture of DNA in a double helix just right, then in some areas, if you section off, you'll see that it looks like an X, kind of where those 2 strands cross there in the middle. Now, Franklin also proposed that the sugar phosphate material is going to be on the outside of the DNA molecule. And that is because they are hydrophilic. If you remember, hydrophilic side chains on amino acids cause them to fold outward so they can interact with water. And the same thing is going on with DNA, is the outer portions or the outer molecules of DNA are going to be ones that are hydrophilic. They can interact with water. And she believed these nitrogenous bases to be on the inside of the DNA molecule because they are hydrophobic. Now, what's interesting about this photo is that it was given, and I put that in parentheses because Rosalind Franklin didn't grant permission for her photo to be shared with Watson and Crick, but somehow they ended up with this photo 51, and they were the ones that basically put together that final model of the structure of DNA with the double helix, with the bases inside making up the rungs of the DNA, and the phosphate sugar backbone on the outside of DNA. So this brings up some ideas about the nature of science and the nature of research, is that research can be fiercely competitive. It was a race to be the ones that published the structure of DNA. But it also shows you that the workings of a lot of researchers contributed to this idea of DNA. So we see that the findings from Griffith aided the research of Avery, McLean, and McCarty. We can see that some of the ideas that Rosalind Franklin discovered, like the structure of DNA with its double helix and the hydrophilic and hydrophobic properties of some of the molecules that make up DNA, aided in Watson and Crick coming up with that final model of DNA. Next, we're going to get into some basics about DNA. And it's important that we also address some of the misconceptions that students have about DNA. Now, you know that you're made of different types of cells, and so a misconception is that each cell has its own specific DNA, and that's not true. The DNA base sequence in each of your cells is identical. So if I wanted to sequence your DNA, I could use a liver cell or I could use a skin cell, and I would get the exact same base sequence. Now, the reason why your cells look different, even though they have the exact same genetic code inside of them, is because these different genes on the DNA are expressed differently. Some genes are turned on in some cells, while others are turned off, and vice versa. So here we have a neuron or a nerve cell, and this is showing you that this gene is being expressed. It's being turned on. But we can see this other gene down here is not being expressed. It's being silenced, or it's being turned off. Whereas in our skin cells, we can see that this top gene that was being expressed in the nerve cell is off, whereas this gene is being expressed. It's being turned on. So the reason why these cells with the same genetic code are different is because some genes are on or expressed in the cell, while other genes remain off, or we say they're silenced. So if we think about this neuron or nerve cell, it contains genes that code for insulin and digestive enzymes, but it should make sense that those genes are going to be silenced or turned off, or they're not being expressed, because a neuron has no need to produce insulin or digestive enzymes. Another idea that we've mentioned is that DNA codes for your proteins, but we're going to get more specific. The bases of your DNA, they code for the amino acids. And I'm not going to say protein. They code for the amino acids in a polypeptide. We said in pre-AP that the genes on DNA code for a protein, and that's not exactly accurate, but we're trying to simplify it for you guys. Actually, one gene codes for a polypeptide. But if you remember the proteins that we built, and they're still hanging up in our room, the ribbon models that we created, is that we had people in charge of one polypeptide chain and its folding, and then other people were folding their polypeptide chains as well. And at the very end, we were talking about that quaternary structure of proteins. Then we put several polypeptide chains together to make a protein. So remember, proteins are usually made of 2 or more polypeptides. So to make most proteins in your body, it takes more than one gene. So if a protein is made of 4 polypeptides, then 4 different genes are needed to code for that one protein. So I just wanted to indicate it's not one gene equals one protein, so I put a slash through there. It's one gene codes for one polypeptide. We've talked on several occasions about the structure of DNA, but there's some big ideas that I want to revisit, and then there's some new information about the structure of DNA. When DNA is uncondensed, or it looks like a string, it is called chromatin. Again, I like to take that I in chromatin and string it out. So remember that chromatin is DNA in stringy form. Now the DNA must be in chromatin form for most of the cell's life, because the DNA has to be replicated or copied in S phase before the cell divides. But the DNA is also continuously coding for proteins, so it's always being transcribed. And the DNA cannot be replicated or transcribed if it's coiled up into chromosomes, like we see here. So again, we talked about that in prophase and mitosis, the DNA coils up into chromosome form. And it coils up into chromosome form just so that the DNA can be moved around during mitosis or nuclear division. But for most of the cell's life, the DNA must be in chromatin form so it can be copied when it needs to be copied and transcribed whenever it needs to be transcribed. Something else to emphasize is that one chromosome is actually one molecule of DNA. So we say that we have 46 chromosomes, or we can also say we have 46 molecules of DNA. Now I want to focus more on chromatin. And you can see here that I have chromatin down here. And chromatin is just DNA that's organized around these proteins that we've talked about before called histones. So here's the histones. And this whole picture down here would be showing you DNA in chromatin form. And you can see that the DNA in chromatin form can even be more loosely coiled or more densely coiled. Now the chromatin that is loosely packed is called euchromatin. And you need to have this memorized. So for me to remember that euchromatin is the DNA that is loosely packed, I remember U rhymes with lu, like loose. The important thing to note about the genes, so we have genes here on the DNA, in sections that are euchromatin, is these genes can be transcribed. these genes can code for proteins. And the reason why this can happen is because the machinery that actually transcribes the DNA, making mRNA. So I'm going to put some machinery in here, and you're going to learn that machinery is called RNA polymerase because it's going to make mRNA. The machinery can get into the DNA, can break it open, and can actually make the mRNA. So let's show some mRNA being made. So the genes that are in the areas of euchromatin are expressed. We're transcribing them, and we can make a protein product from them. Now, chromatin that remains tightly coiled is called heterochromatin. And the genes in this section of DNA are silenced. They are not expressed. So no transcription is taking place in this area. So no mRNA is being made. That means no proteins are going to be synthesized from these genes. And we say that these genes are silenced. So all the genes in this area of the DNA molecule are off. They're not being expressed. I think this is about the third time where we've highlighted the structure of DNA. So again, I'm just going to hit the highlights. DNA is made of monomers that are called nucleotides. DNA is made of carbon, hydrogen, oxygen, phosphorus, and nitrogen atoms. Nucleotides have three parts. There's the phosphate group, that 5-carbon sugar, and that nitrogenous base. It's key that you remember that DNA contains carbon, hydrogen, oxygen, phosphorus, and nitrogen in its nucleotides. So when we talk about tagging molecules, you'll understand why we tag molecules with different tracers compared to others. The sugar phosphate backbones make up the outside of the DNA molecule. They're joined together by phosphodiester bonds. In between the bases, we find those hydrogen bonds. And remember that C and G have 3 hydrogen bonds. A and T have 2. The purines are pure ags, adenine and guanine, and they're the double rings. Single rings are those, and those pyrimidines are the thymine and cytosine bases. And we say each rung. So what I mean by a rung is this is one rung of that DNA molecule. And you can see that every rung contains that single and double ring base. And this ensures that the diameter of DNA is constant all throughout the molecule. And then we mentioned the ends of the nucleotides, and we have the 5' end versus a 3' end, and we can see that this is the 3' end, this is the 5' end, because the strands of DNA are anti-parallel to one another. Now, another idea I want to revisit is the fact that anytime you join monomers together, so you should be able to look at this and you understand this is a monomer of DNA. And you should be able to tell that because there's a phosphate group, and we see a sugar right here. And then lots of times they'll put nitrogen atoms here on those bases, so you can recognize that's a nitrogenous base. So we have one monomer, and here's another monomer. Anytime we join 2 monomers together, it's a dehydration synthesis reaction, or we can call it a condensation reaction, because we're pulling water out or dehydrating it to join 2 monomers together. So you can see here is where these 2 monomers are going to be joined together. Out comes water, and we can now see that 1 oxygen and 2 hydrogens are now missing from this area of the molecule because we removed them forming water. Now you're going to see all kinds of practice problems that requires you to have an in-depth knowledge of the structure of DNA. And here's an example of one of these problems. See if you can pause and solve. Problem says, what % of the DNA bases are thymine if you determine that cytosine makes up 20% of the DNA molecule? So you have to remember the base pairing rules, that A goes with T and C goes with G. So if we determine that cytosine makes up 20%, there should be a 1-to-1 ratio of cytosine to guanine because they're always paired together. So 20% of the DNA should be guanine. That leaves 60% left. That's going to be made of the adenine and thymine. So if you split that so there's a 1-to-1 ratio, 30% of the bases should be adenine and 30% of the bases should be thymine. So that's an easy problem if you've seen it once. Otherwise, it can be a little bit tricky to those that haven't seen this type of problem before. Now the next lecture is going to be about protein synthesis. And before we go into that, I want to talk about other nucleic acids. So we have DNA, but then we also have RNA, and there's different types of RNA. There's messenger RNA and transfer RNA. And you're going to learn there's RNAi molecules, and there's different types of RNA molecules. And we also talked about ribosomal RNA, so let's revisit those quickly. So RNA is a single strand of nucleotides. And it has 4 bases as well, but it has adenine, guanine, cytosine, and U's. Those U's stand for uracil. There's no thymine in RNA. Now, RNA stands for ribonucleic acid because it's being named after the sugar. So the sugar in RNA is ribose. The sugar in DNA is deoxyribose. And you should know that ribose and deoxyribose are sugars because they end in ose, like many other sugars do. Now, there's different types of RNA. We're going to talk about messenger RNA is the nucleic acid that's made in transcription. It's a copy of the gene off of the DNA. And you're also going to learn that some things have to happen to this mRNA molecule. It has to be processed. So after we make this mRNA, you're going to see that the cell is going to add 2 things to it. It's going to put a cap on it. We can see that cap right there. And it's also going to get a tail. And that tail is made of nucleotides with adenine on them, repeated over and over. Sometimes there's about 200 adenine nucleotides put on the end of an mRNA molecule. And we're going to talk more about the cap and the tail, but I just wanted to show you a diagram of mRNA that you're not used to. And if you see a molecule that has a cap and a tail on it, then you should pick up that's an mRNA molecule. We're going to talk more about tRNA molecules, again, another type of RNA. tRNAs are used in translation at a ribosome, and tRNAs look like a T. So I think it's easy to identify a tRNA because it kind of looks like a T, like you see over here. And if you see a molecule that has an amino acid on one end and an anticodon on another, then you should pick up that you're looking at a tRNA molecule. Now, the next type of RNA I'm going to mention is a type of RNAi molecule. So I'm going to indicate there's molecules that are called RNAi molecules. And one type of RNAi molecule is a microRNA. And we're going to learn more about microRNAs, but I'm just kind of listing the different types of nucleic acids that you're going to have to be familiar with in this unit. And then finally, let's mention ribosomal RNA. Ribosomal RNA is found in ribosomes. So if you have ribosomal RNA plus some proteins, that equals a ribosome.